Hi, my name is Albert Lin, and this is my thesis, Bringing Butterflies and Moths Back to Life, Recreating Lifelike Poses in Museum Specimens. In this presentation, I will go over my research rationale, the data sources that I used, the course of development of my app, and the results of the study. So, why butterflies? Butterflies are an important part of the ecosystem as they are involved in many layers of the food web. As larvae, they are important herbivores, and as adults, they perform dual roles as both pollinators that ensure the plant's own reproduction, as well as providing food for mid-level predators. Butterflies and moths are also important to people culturally and economically. In terms of agriculture, they're responsible for a lot of pollination, especially of wild foraging plants, as well as being the primary source of silk for our textile industry. They are also pests and are responsible for a lot of crop destruction in their larval forms. Tracking populations is therefore an important part of biodiversity and ecological health. But to get good measurements, it requires a lot of data points over time. This need for big data has caused some studies to search for new ways to obtain and process that data. One is the use of citizen science, which leverages the greater population to help with gathering sightings of butterflies in an area and tracking those changes over time. The other is the use of machine learning to be able to process much more data and more quickly than humans can. iNaturalist is a citizen science initiative aiming to gather field images taken in the wild of living specimens. Users then also vote on what they think is the correct species, eventually creating a label via consensus. This allows a lot of images to be labeled with a species, as the quantity of users is much greater than the expert researchers at scientific institutions. But this also introduces inherent biases toward more well-known species, with an equal weight given to an expert's vote as an amateur enthusiast. These images are then also processed into a machine learning model that is able to identify photographs of moths that are submitted to their model. In contrast, BOLD, which stands for the Barcode of Life Data System, largely has submissions from experts. This is because the principal data for BOLD is the genetic barcode of each species, which is a record of DNA sequences unique to the species. As such, most of the images are pulled from museum collections, many of which have the wings pulled forward unnaturally to best display the entirety of the wings when they are mounted with a pin. There is a research gap between studies that have used museum images and studies that have used citizen science to inform machine learning models to identify species. Therefore, the study aims to bridge that gap by transforming museum images into poses that better match the poses of living specimens. While it is possible to make those changes manually with photo editing software like Photoshop, the point of using machine learning systems is to be able to process large quantities of data. Therefore, it seemed like the best way to make this work is to automate the process as much as possible. The butterfly image conversion algorithm was conceived to break down the process into steps to inform the programming. The resulting image would then be tested against a machine learning model that is trained on iNaturalist images and labels. The beginning of the project involved quite a lot of trial and error before finding a solution. The first step was to convert the images to grayscale. Color images have additional data for the RGB channels, which still causes complications for a lot of computer vision methods. Canny edge detection was initially used, as it had been suggested by the Copilot AI when I was initially doing research. However, this ended up not being very usable, as there were too many gaps in the detected edges. My attempts to plug those gaps with radiating pixel searches often failed and caused the program to hang up. Instead, I used a binary mask to find the edge, taking advantage of the contrast between the background and the butterfly in many images. However, this was not universally applicable to all images from our bold source, and so it caused many failed conversions as well. Thankfully, enough of it worked often enough that I was able to move forward and produce a substantial data set for testing. The binary allows us to make a continuous line for the outline boundary. The next step is to break the butterfly into parts. As I had pre-stored the images to only consist of images of the butterflies in their canonical pose, I can safely assume that the specimen would exist in the middle of the picture. From there, a bounding box expands until it reaches the extremities of the butterflies, thus telling the program the area the butterfly is in. From there, the important vertices are found on the contour to mark out where each body part is. This also required additional assumptions based on the typical butterfly body plan, so this is another point of failure as atypical wing configurations can cause scrambling of the final image or outright failure. Once the extreme vertices have been found, however, the contour can then be split up into the relevant body parts in preparation for segmentation. With the boundaries of the segments ready, the program can then extract the pixel information from those areas to store them temporarily into arrays. These arrays are then transformed to be able to rotate about the vertex where the wing meets the body. This involves expanding the image and translating the extracted pixels so that the pivot point is in the center of the new image. Applying the rotation, then collapsing it back down to just the extracted pixels after they have been rotated into the desired position. The angle I settled on for the wing rotations was a result of a lot of trial and error with the mathematics, and while it subjectively looks quite good from many of the output images, the wing angles don't always match the living specimen photos, especially for the back wings. In the end, the program did successfully manage to create many images for the test data set. However, the results did not improve identification compared to the unmodified images. Part of it may have been the introduction of digital artifacts from mistakes in the programming. So while I accomplished the goal of automatically changing the wing pose, the introduction of those artifacts may have hampered the identification of species due to sometimes significant changes to the apparent pattern of the wings. In addition, there was a high failure rate with moth species. Part of this was due to their more neutral coloration, resulting in a lack of contrast with similarly neutral backgrounds. Cryptic patterns also threw off computer vision, and the increased fuzz of many moth species compared to butterflies made finding the outline difficult. This area also had performed more poorly in previous studies, and the inability of our program to process those species does not provide an improvement. This graph shows that the species I was able to successfully create an output for were already species that the machine learning model already had little trouble identifying. However, even with the handicap of digital artifacts, there were a significant number of instances where the output image from our test dataset performed better than the original. I do think there is some promise to the method, but the inability to improve on species that were already poorly performing may require a new approach to address that shortcoming.